Hello, my name is Bill Andrews. I'm a medical research scientist that has been studying aging my entire life. I've developed a great understanding of what aging is, why we age, how we age, and ways we might be able to interfere with aging, something that I often refer to as curing aging. My main goal is actually to cure my own aging. Now, there's a lot of theories on why we age, but is there a kingpin that controls all of them? The answer is we just don't know yet. But the most likely kingpin, if there is such a thing, is telomere shortening. No matter what else we do to try to solve the aging process, nothing else is ever going to work unless we also solve the telomere shortening problem. And solving the telomere shortening problem might be the only thing we have to do, because telomere shortening might control all the other theories of aging. Telomere shortening is the only theory of aging that can actually say this. For those of you that already know about telomere biology, you know that there's a lot of controversy in the field. And I've addressed all those concerns and issues in a video on my website called Everything About Telomeres. My website is www.sierrasci.com. That's short for sierrasciences.com. It's a 2-hour video, but what's really nice is it has a table of contents that shows all the different issues you might be interested in, and all you got to do is go to that part to listen to any concern or issue that you have. And again, that's it. It's called Everything About Telomeres, and it's at my website, www.sierrasci.com. So what's a telomere? To answer this, we need to zoom in on a human being. We find that a human is made up of 50 to 100 trillion cells, and every cell contains a nucleus, shown in the middle of each of these cells. If we zoom in on one of these nuclei, we find that the nuclei contain chromosomes. The chromosomes are where our genes are, that give us our hair color and our eye color, etc. If we zoom further and zoom in on one of these chromosomes, we find that a chromosome is made up of 2 arms, a top arm and a bottom arm. And inside of each of these arms is a long DNA molecule. It's a long string of beads. The beads are called bases. The typical DNA molecule inside of a chromosome has about 100 million bases. And it's so long that it's coiled up like a slinky inside of each of these arms. Now I want you to think of this DNA molecule as a shoelace. And you know at the tips of your shoelaces you have these caps called aglets that protect your shoelaces. Where DNA molecules inside of our chromosomes have the same thing, except they're called telomeres, shown here in yellow. Telomeres protect our chromosomes, and they're the aglets of our chromosomes. If we zoom in on one of these telomeres, we find that a telomere is about 15,000 bases in length, at least when we're first conceived. And this is where all the troubles begin, because as soon as our cells start to divide, each and every time our cells divide, our telomeres get a little bit shorter. When we're born, our telomeres are already down to 10,000 bases. Remember, when we were first conceived, our telomeres are 15,000 bases, but when we're born, they're already shortened down to 10,000 bases. But that's okay. That's like cutting 1/3 of your shoelace off. Your shoelace is still fine. But the problem doesn't end there, because we have still a lot more growing to do, a lot of wounds to heal, a lot of immune responses to pathogens, and when our telomeres get down to 5,000 bases, we die of old age. Our cells lose the ability to function, and we die. Let me go over that again. We're conceived at 15,000 bases, we're born at 10,000 bases, and we die of old age at 5,000 bases. There's absolutely nothing we can do about that yet. No matter how well we eat, no matter how much we exercise, no matter how much we do everything our doctors tell us to do, we cannot prevent this shortening. It's the only real clock of aging that's ever been discovered in humans. And if you do the math, you can calculate that the rate of telomere shortening predicts that we have a theoretical maximum lifespan of 125 years. And I need to mention that Practically every disease you've ever heard of has now been connected to the length of your telomeres in the scientific peer-reviewed literature. These diseases of short telomeres are called telomeropathies. So it's not just aging. Bottom line is, bad things happen when telomeres get short. So this is where I entered the picture about 30 years ago. At that time, we knew nothing about cause or effect. I mean, did telomere shortening cause aging, or did aging cause telomere shortening? We just didn't know. But I wanted to find out because I was obsessed with trying to find a cure for aging, and this seemed like a good place to start. But first, let me just discuss why I'm so obsessed with curing aging. I'm not afraid of dying. I'm afraid of missing out on all the great things that are going to happen in the future. One of these days, we're going to find intelligent life out there, and when that happens, I want to be there. But it's not just that. I love living. And I would hate the idea of living becoming any less lovable for me or anyone. Living is the greatest thing that ever happened to us. Let's just not throw it away because we think it's meant to be. I didn't mention this before, but I run a biotech company called Sierra Sciences in Reno, Nevada, USA. I founded the company in 1999, which was 23 years ago. As I mentioned, I'm a medical researcher. I got my PhD in molecular genetics and evolution, and I've been in biotech now for 40 years. In 1997, I was awarded second place for National Inventor of the Year in the United States for my cancer research. So I do know quite a lot about cancer. I'm also an ultramarathon runner because I believe that endurance exercise is one of the best things you can do to delay the aging process. I've been featured in several documentaries. 3 listed here are The Immortalist that I co-starred with Aubrey de Grey. It almost won an Oscar at the Academy Awards. I was also in The High, and I'm going to be in an upcoming documentary called Longevity Hackers, which should be out later this year in 2022. I also co-authored a few books. One is called Curing Aging, and the other one's called Telomere Lengthening. And I highly recommend that you read both of them. A lot of my ideas in aging and biotech in general are unique. And that's because throughout my career, I've picked up a lot of skills and tools listed in this slide that has enabled me to draw my own conclusions. As a result, I've had a very successful biotech career, playing key roles in a lot of the biggest biotech breakthroughs in the past. So let's get back to telomeres. How are we going to keep telomeres from shortening? Well, we begin with the observation made a long time ago that telomeres don't shorten in our reproductive cells. I had mentioned before that every time a human cell divides, the telomeres get a little shorter. But that's not true in our reproductive cells. And that's important because if they did shorten, our children would be born with shorter telomeres than we have. Their children would be born with shorter telomeres than they have. And we would have been extinct as a species millions of years ago. So what is it that our reproductive cells have that prevents their telomeres from shortening? Well, I led the research in the early 1990s where we discovered the enzyme telomerase, which is found in our reproductive cells and is the reason why our reproductive cells don't have telomere shortening. This is a cartoon. The green squiggly thing is the DNA molecule showing the region called the telomere. And at the very tip of the telomere is this factory-looking thing, which is an enzyme called telomerase. So every time a reproductive cell divides and the telomeres get a little shorter, the reproductive cells have this enzyme telomerase that comes in and re-lengthens the telomeres by adding bases, shown here in green. So the telomeres don't shorten in our reproductive cells. Well, we then did a bunch of experiments with telomerase, putting telomerase into cells and lengthening the telomeres, and we found that, one, we could reverse aging in human cells in vitro. First time that it's ever been done. we had exceeded something called the Hayflick limit, where it had been known since the 1960s that human cells can only divide a certain number of times. When we put telomerase into the cells, they no longer encountered that Hayflick limit. The cells kept on growing. In fact, they became younger by every method of measurement we had available to us. Then we grew skin on the back of a mouse and put telomerase into the skin cells and showed that the human skin got younger on the back of a mouse. And again, the human skin got younger by every method of measurement that was available at the time. So then we sent the gene for telomerase to a lab in Harvard where they created engineered mice. These mice were allowed to grow old, and then when they got old, they were fed a substance that caused them to start producing telomerase in all their cells. And what they saw was that these mice got younger in every way imaginable. So again, telomere lengthening has demonstrated reversal of aging by 3 different ways. And no other theory on aging has ever been able to do any one of these things. So why do only our reproductive cells produce telomerase? Why don't all of our other cells produce telomerase? If our reproductive cells produce it, we must have the gene for telomerase in all of our cells. Why is it that our other cells don't produce telomerase? Well, it's all about gene regulation. Telomerase is produced in our cells by the telomerase gene. Next to every gene is a regulatory element. But think of this as a dimmer switch instead of like a regular on-off light switch. In our reproductive cells, that switch is turned on, so our reproductive cells produce telomerase. But in all the other cells of our body, there's a protein that binds to that switch and shuts it off. That protein is called a repressor. Now there's 2 ways to fix this problem. One is to find a way to dislodge that repressor, and that's called gene induction. The other way is to deliver a new gene that has a regulatory element that the repressor doesn't bind. That's called gene therapy. Now we do both approaches at Sierra Sciences to try to turn on the telomerase gene. The first method I described, gene induction, involves testing a lot of chemicals and natural products, looking for anything shown in green here that can bind to the repressor and dislodge it, thus turning the gene on. Now, this is very intensive. It involves a lot of our scientific disciplines to just design chemicals to be tested. Once we find a chemical or a natural product that we want to test, we send it into our high-throughput robotic screening system to test it in telomerase assays. When we find chemicals and natural products that actually do turn on the gene to some extent, that hit is sent back to those scientific disciplines where they then design even better hits given the new information that they have. And this is back and forth, getting stronger and stronger and stronger hits. So far, we have tested over 350,000 different chemicals and natural products in our telomerase gene induction assay. And we have found over 900 different chemicals or natural products that will induce telomerase to some level. And we've been able to divide these into 39 different families, which has been very informative to the medicinal chemists that are designing new chemicals. At this time, the strongest hit that we have discovered produces about 16% as much telomerase as what we think we need to completely stop telomere shortening. And we plan to continue doing this screen until we find something that exceeds 100%. It costs us a little over $1 million per month to do this research. But when we find chemicals or natural products that are going to work, it's only going to cost us about $1 to produce enough to treat one person once. The other method that I mentioned for producing telomerase in a human cell is telomerase gene therapy. And our telomerase gene therapy already produces more telomerase in a human cell than in what is needed to stop telomere shortening. It in fact lengthens telomeres. I want you to think of a human cell as a large soap bubble. And I want you to think of the gene therapy as a smaller soap bubble. And inside that smaller soap bubble is the gene for telomerase. Well, we all know that when two soap bubbles come together, they fuse, and the contents of one goes inside the other. Once that gene is inside the human cell, it then produces telomerase. And because we've engineered this gene so that it's very strong, it produces a lot of telomerase. Now, this protocol costs us approximately $1 million per person, depending on how many tests we want to do, how many doctors are involved, what hospital costs, etc. Now, I'd already mentioned that practically every disease you've ever heard of is affected by the length of your telomeres. So it means that there's a lot of telomeropathies that could be treated with this gene therapy. We've already written many telomerase gene therapy clinical protocols that are about 140 pages each. First one we wrote was just for aging in general. We've also written them for Alzheimer's disease, critical limb ischemia, and cardiomyopathy. We're presently writing a clinical protocol for demyelinating disorders such as multiple sclerosis, and we have many more coming in the future. Sierra Sciences is not a clinical testing facility, so we plan to partner with BioViva, the leader in gene therapy technology for treating aging, that is headed by Elizabeth Parrish. Elizabeth Parrish has partnered with Dr. Jason Williams, who I consider to be the leading cancer doctor in the world. Together, they have already used gene therapy to deliver the Klotho gene, the follistatin gene, the PGC1 alpha gene, and telomerase. And you can learn more about BioViva at bioviva-science.com. Gene therapy does come with risks, but you have to ask yourself, when is the risk from no treatment greater than the risk from treatment? Sometimes the most serious risk to the patient is actually not treating the patient at all. Some diseases, such as Alzheimer's, are 100% fatal, so the risk from no treatment is very, very high. The same is true for a healthy 89-year-old. A healthy 89-year-old actually has an 11% chance of dying by the time they turn 90. So again, the risk from no treatment for an 89-year-old is very high, and getting treated might be the lesser risk. And lastly, I want to mention that a lot of people believe that telomerase will cause cancer. And there is no reason to believe that that might be true. With my cancer background, I strongly believe that telomerase will actually decrease the risk of cancer. And to hear more about it, you can read my book, Telomere Lengthening, that's available at Amazon.com, where I have a whole chapter describing why telomerase will not cause cancer. Then also on my website, there's a video called Tokyo Presentation 2017, where I spent about 45 minutes going over in excruciating detail the reasons why telomerase will not cause cancer and will actually do the opposite. So go to that video and start watching at about 25 minutes and 25 seconds. So I've described two approaches that we're taking to produce telomerase in human cells, lengthen telomeres, and cure aging. Not only in humans, but also our pets. Presently, both approaches are on hold while we await funding, so I can't give you a time frame as to when this research will be done. But one way or another, we're going to get the work done, and I hope to see you there. Thank you.